Welcome to this lecture on transposable genetic elements. Imagine if the paragraphs in a book could detach themselves and jump to entirely different pages while you were reading it. In the world of genetics, this actually happens. Today, we are going to explore these fascinating jumping genes, understand how they move, and discover why they are absolutely crucial to the evolution of life on Earth. To begin, let us think of your DNA as a massive, thousands of pages long instruction manual for building a human body. Now, imagine that certain paragraphs in this manual suddenly cut themselves out and paste themselves into completely different chapters. If a paragraph lands in the middle of a crucial instruction, it might ruin the sentence and cause a catastrophic mistake. But on the other hand, it might accidentally create a brand new, useful instruction that makes the final product even better. These jumping paragraphs are what we call transposable elements, constantly rewriting the genetic story. Now that we have a conceptual picture, let us look at the formal definition. Transposable genetic elements, often referred to simply as transposons or jumping genes, are specific sequences of DNA that possess the astonishing ability to change their position within a genome. This groundbreaking phenomenon was discovered by the brilliant geneticist Barbara McClintock in the 1940s. While studying the genetics of maize, or corn, she noticed that certain genetic traits were turning on and off in ways that could only be explained by genes physically moving around the chromosomes. This completely changed our understanding of genetics. Understanding how these elements were discovered leads us to ask, how exactly do they move? Transposons utilize two primary mechanisms. The first is the cut and paste mechanism, where an enzyme physically excises the DNA sequence from its original location and inserts it somewhere else. This leaves a gap behind and can disrupt genes at both the exit and entry points. The second is the copy and paste mechanism. Here, the DNA sequence is transcribed into RNA, reverse transcribed back into DNA, and then inserted into a new location. This leaves the original copy intact, effectively doubling its presence in the genome. Let us visualize these two mechanisms side by side to make the difference crystal clear. On the left side of this diagram, you see the cut and paste mechanism used by DNA transposons. Notice how the blue transposon is completely removed from the donor DNA, leaving a visible gap before it integrates into the target DNA. Now look at the right side, showing the copy and paste mechanism used by retrotransposons. The original orange transposon stays exactly where it is. Instead, it creates an RNA intermediate, which is then converted back into DNA and inserted into the target site. This fundamental difference explains why retrotransposons can rapidly expand genome size. Having seen how they multiply, you might be wondering why our bodies tolerate DNA that randomly jumps around. The truth is, transposable elements are incredibly important drivers of genetic diversity and evolution. Without them, genomes would be static and unable to adapt to changing environments. In fact, nearly half of the human genome is made up of these transposable elements. They act as a vast reservoir of genetic material that evolution can occasionally repurpose for beneficial functions, such as the development of the mammalian placenta. Furthermore, modern scientists have harnessed these jumping genes as powerful tools for genetic engineering. Because the copy and paste mechanism leaves the original gene intact, it leads to a fascinating mathematical consequence: exponential growth. We can model this accumulation using a standard exponential growth formula. The formula is n sub t equals n sub 0 times the quantity 1 plus r raised to the power of t. Here, n sub t represents the total number of transposon copies present after a certain time. N sub 0 is the initial number of copies at generation 0. The variable r is the transposition rate, meaning the probability of a successful copy event per generation. Finally, t is the number of generations. If the rate r increases, the genome size will expand much more rapidly. Now that we have our formula, let us walk through a practical example to see it in action. Suppose a newly discovered yeast species has an initial count of 100 copies of a retrotransposon. Geneticists determine the transposition rate is 0.05, or 5%, per generation. We want to find the total copies after 10 generations. First, we substitute our known values into the formula. The expression inside the parentheses becomes 1.05, which is our growth factor. Next, we raise 1.05 to the power of 10, which gives us approximately 1.62889. Finally, multiplying this by our initial 100 copies gives us roughly 163. This shows how quickly genomes can expand if transposons are left unchecked. While retrotransposons expand the genome, the cut and paste transposons present a different kind of challenge. This brings us to the danger of gene disruption. When a transposon jumps to a completely random location, there's a risk it might land right in the middle of an essential gene. If this happens, it can disrupt the gene's function and cause a lethal mutation. The likelihood of this catastrophic event depends heavily on how much of the genome is actually made up of essential genes compared to non-essential DNA. Because of this constant threat, organisms have evolved complex silencing mechanisms, like RNA interference, to keep these jumping genes locked down and protect their vital genetic instructions. To quantify this danger, we first need to determine exactly how large the target area is for a potential disruption. We calculate the total target area of essential genes using a simple multiplication formula. T sub genes equals G times L. In this equation, T sub genes represents the total number of base pairs in the genome that belong to essential genes. The variable G stands for the total number of essential genes present in the organism. The variable L represents the average length of each essential gene, measured in base pairs. If an organism has more essential genes, or if those genes are longer, the variable T sub genes will increase, creating a larger target for transposons to hit. Let us apply this target area formula to a concrete biological example. Imagine a fruit fly genome that contains exactly 15,000 essential genes. We are given that the average length of an essential gene in this fly is 2,000 base pairs. We want to find the total amount of DNA dedicated to these vital instructions. By substituting our values into the formula, we multiply 15,000 genes by 2,000 base pairs per gene. This calculation yields exactly 30 million base pairs. This 30 million base pair region is the critical target area where a transposon landing would cause a dangerous mutation. Now that we know the size of the target area, we can determine the actual likelihood of a disruption occurring. We model this using a basic geometric probability formula, assuming the transposon has an equal chance of landing anywhere in the DNA. The formula is P equals T sub genes divided by S. Here, P is the probability that a jumping gene lands inside an essential gene. T sub genes is the total target area we just learned how to calculate. The variable S represents the total size of the entire genome in base pairs. If the overall genome size, S, is very large compared to the essential genes, the probability P decreases, which actually protects the organism. Let us finish our fruit fly example by calculating this final probability. We already found that our target area, T subgenes, is 30 million base pairs. Now, suppose the total size of the fruit fly genome, S, is 150 million base pairs. We want to find the probability of a single random transposition event disrupting an essential gene. We set up our formula by dividing the 30 million target base pairs by the 150 million total base pairs. The calculation simplifies to 0.20. This means there is a 20% chance that a random jump will cause a lethal mutation, highlighting exactly why cellular defense mechanisms are so critical. We have covered a lot of ground today, so let us review the most important ideas. First, transposable elements are mobile pieces of DNA that independently move around the genome using either cut and paste or copy and paste mechanisms. Second, while we calculated the very real danger they pose by disrupting essential genes, remember that they are also a massive source of evolutionary innovation. They provide the genetic diversity that allows species to adapt and survive over millions of years. You have now mastered the fundamentals of jumping genes, proving that our DNA is never just a static blueprint, but a dynamic, ever-changing landscape.